So this recording may be available by request, but that's mainly for our own purposes. Okay. So the webinar will be 30 minutes of presentation from Mr. Drew Horton, followed by 30 minutes of presentation from Dr. Misha Kwasniewski, followed by 30 minutes of question and answer. So during all the webinar, you can type your question in the Q&A box or the chat box. If you don't see the Q&A box or the chat box, they should be accessible when you click on the three dots on the bottom of your screen. So make sure you have access to that and type any question you have during all the webinar, and everything would be answered at the end. And if we don't have time, I think we would have time, but just in case, if we don't have time at the end, don't worry, we will try to answer you by email. Also, one more thing is that at the end of the webinar, you will receive an email with a link for a survey, because we are always looking for feedback to improve ourselves for the webinar that we are offering you. So the survey, please take just 2 to 5 minutes to complete the survey at the end. So on that, as I explained you, so we start by a presentation from Mr. Drew Horton from the University of Minnesota, followed by a presentation of Dr. Misha Kwasniewski from Penn State University, followed by the question and answer. And so I would like first to thank you both for agreeing to participating and presenting in this webinar today. So I'm gonna start by making a short introduction about Drew Horton. So Drew, I'm gonna read, sorry. So Drew Horton has been active in the wine industry for over 20 years, having started as a wine salesman. He then worked the wine production ladder from cellar rat to cellar master, and eventually winemaker starting in 2004. With a BA in anthropology and early career experience working in kitchens, Drew brings a unique perspective at the intersection of food, culture, and of course wine. He produced award-winning wines in Santa Barbara County, California, before moving to Minnesota in 2010, where he began working with cold hardy hybrid grapes as founding winemaker for Chankaska Creek Winery. In 2015, Drew moved from commercial winemaking to research winemaking and providing outreach as a field specialist enologist for the Midwest Grape and Wine Industry Institute at Iowa State University in Ames, Iowa. As enology specialist for the University of Minnesota's Grape Breeding and Enology Project, Drew produces nearly 100 microlots of research wine and ciders annually. As well, he engages with and provides university and extension resources to winemakers across the state. Drew, can you mute yourself just quickly? This includes troubleshooting, planning, and evaluating winemaking processes for Minnesota's farm wineries. Drew has broad experience and expertise in winery design and equipment, fermentation techniques, barrel aging, filtration, and blending. Additionally, Drew offers courses and workshops to appeal to the needs of winemakers, from amateur to professional. Horton says, "I'm a hands-on winemaker, always looking for the best practical solutions to solve wine and winery needs and issues." So today, Drew is going to present and discuss the tools available for and the timing on how to adjust acidity in the must and wines, with a specific focus on interspecific hybrid grapes and wines. So I'm gonna stop sharing my screen. I will stop my video probably as well. And Drew, you can start whenever you want.
Do you need to make me a presenter? No, I have share here. Okay. And there's that.
You should be
And
good.
there we go. Can you see my screen?
Yep, you can put the slideshow. Yep,
Okay.
all good. Thank you.
All good. Okay. Thank you, Aude. Thank you all. I'm glad to be here. Never in my wildest dreams, 10 years ago when I left California, did I think I'd be here doing what I'm doing now. But I do love what I'm doing. I'm staying here in Minnesota. I love working with these new grapes. Of course, as you all, or most of you are aware, most of these northern climate grapes have some challenging chemistry. They have low pHs and high total acidity. And I can assure you we're working on that at the grape breeding program at the University of Minnesota. We're trying every day to come up with new grapes that have improved cold tolerance, improved disease resistance, and more moderate acidity. Itasca is, of course, the big victory we've had recently. The very first Itasca grape wines are coming out this year. And we just picked last week a late harvest bit of Itasca that was still hanging. And the Brix was 27.5 with pH of 3.29 and only 5.3 grams of total acidity. So we're getting there, and I can assure you I made a lot of new red wine batches this year. I was just telling Misha and Aude, I've now completed 107 grape wine batches this year, and haven't even gotten into the ciders. I'm fortunate that we can freeze the cider apple juice until I'm done with the grape harvest. And sometime when things slow down in January, I'll move on to thawing and working with that cider juice. We're looking at University of Minnesota apples, which are, of course, were bred to be dessert apples, and we're seeing if they are also possibly appropriate for cider production. All right, enough about that. We'll move on with these practical high acidity winemaking strategies for the Midwest. You don't need to have a degree in chemistry to understand winemaking, but you should have a basic understanding of pH and total acidity in practical winemaking. Don't be afraid of pH. Hopefully, I can help clarify any confusion or questions you may have about dealing with pH and how to interpret your pH readings. Of course, maybe not of course, but pH and total acidity or total titratable acidity, they are related, but it's not a direct relation. That's not a one-to-one correlation. Wines and musts on the high end of the scale, let's say 3.65 and higher, usually tend to be lower in acidity. Lower pH usually means higher acidity. But as I say, it's not a one-to-one correlation. They're related, but not the same. But we do have to be aware of both of them. A little refresher or maybe a primer for some of you on pH. In chemistry class, we learned that pH is the inverse log of hydrogen ions in a solution, which will confuse you right off the bat. You have to know what an ion is, and you have to know what an inverse log is. But let's just break that down, make it a little more simple. pH is a logarithmic scale from 0 to 14. Water is neutral. It's in the middle of the scale. It's neither acidic nor basic or alkaline. And it is a logarithmic scale, just like the Richter scale for earthquakes in California. For example, a wine or a must with a pH of 3.0 is 10 times more acidic or has 10 times more hydrogen ions than something of a pH of 4.0. Typically, in most winemaking, we're hopefully dealing with 3.1, the range of 3.1 to 3.65. I'm sure some of you out there have worked with lower pHs and higher pHs. And this is part of the reason for our talk, is to figure out if we get a wine that is a very, very low pH and high acidity, how can we mollify that? How can we tame that down a bit? And in winemaking, pH is really important regarding microbial stability. If your pH is too high, if it's over 3.65 or over 3.7, the wine is more prone or more likely to spoilage organisms. So pH is about microbial stability. It's about how the wine feels in your mouth, its structure. It has some very important properties regarding color retention and ageability as well. Lower pH wines will tend to age longer, whereas higher pH wines won't age quite as long. TA, or as scientists call it, TTA, total titratable acidity. We abbreviate it as total acidity in practical winemaking. It's really an empirical measurement of all the acids in a solution, usually measured in grams per liter or percentage. You just move the decimal point one way or the other. The main point is that the higher the total acidity in solution, the more tart or crisp or vicious the wine will feel on your tongue. The primary acids in wine are tartaric acid and malic acid. There are also trace amounts of many other acids: citric, lactic, acetic, hopefully not too much acetic, succinic, nicotinic, all these other trace acids. But really, we're concerned with tartaric and malic acid in wine. And so people, after I say this to people and give this little lecture, they say, well, what does that all mean? What is the bottom line? And in my mind, as a winemaker and a wine taster, pH is really about the quality of the acids in solution and how it's going to feel on your tongue, whereas total acidity is really just about the bottom line quantity of acids, if you will. Again, there's a correlation between them, but to sort of keep them clear in my mind, I like to look at that sort of thing. pH is about the quality of the acids. All the science I've got today, and I hope that wasn't too painful. I am a practical, close to the ground, experienced practical winemaker, and I remind you all, it always breaks my heart when a winemaker just picks up the phone and calls the grower and says, hey, send me that Frontenac when it's ripe. I encourage all of you, whenever possible, to get out in the vineyard. This is one of my favorite quotes: the best thing for a vineyard are the footprints of the winemaker. It's very important to, especially after veraison, during fruit ripening, that you get out there and physically look and ask questions and touch and taste and smell and use all your senses out in the vineyard, because there's a lot more going on in the vineyard. Though we're concerned with the numbers, with pH and TA and bricks and all the rest of it, it's not just doesn't just come down to the numbers. There's a lot of other things going on that a winemaker needs to know. So as I say here, for me, winemaking starts in the vineyard. Wouldn't have winemaking without a vineyard, of course. And what's going on in the vineyard, how they're training the vine, pruning the vines, how they're managing the canopy, how long they're letting the fruit hang, all of those viticultural practices can have a major or minor, but will have effects on pH and TA. I always encourage an active collaboration between viticulturalists and winemakers. They really are two wings of the same bird. There are a few of these interspecific hybrids, these northern grapes, that have more moderate acidity and more moderate pH. Some of the Swenson varietals like Prairie Star, St. Pepin, and Brianna, Edelweiss, those have more moderate acidity and pHs. So I love to encourage blending. I'll mention it later, but it's not rocket science. If you've got a high acid wine, blend it with a low acid wine. That's one way to balance it out. So I don't focus just solely on the University of Minnesota varieties. I look at try to be aware of all varieties, and not just the Swenson ones, the French hybrids, Chambourcin, those types of grapes as well. To me, blending is a very powerful tool. We'll get into more of that later. But to remind ourselves of what's going on in the vineyard. We have concentration on the what is that, the X or the Y? That's the X-axis, I believe, and then time on the Y-axis. You mathematicians can correct me later. What's going on through time, of course, after veraison is the acidities are falling while the sugars are rising. We always try to find that perfect point in time when everything comes together. The grapes look great, they smell and taste great, their sugars are high enough, their acids are starting to go down. So decision of when to pick is an essential decision in how to deal with these high acid grapes. Can't tell you how many times I got a call in August and early September, people asking me, hey, what bricks do I pick my Marquette? What bricks do I pick Frontenac at? And as I've just said in previous moments, it is more than a number. You want to look at the grapes. You want to look at the vineyard. You want to look at the vineyard as a whole. You want to look at individual clusters. You want to look at the ripeness, not just of the grapes, but of the skins of the grapes, the ripeness of the stems and the seeds, all of these types of things. You and the grower and viticulturalist, of course, want to be aware of what's going on in terms of crop load, in terms of your disease and pest pressure on the vineyard. If you've got a ripe crop hanging and the birds are just mercilessly attacking it, you may have to pick it earlier than you would like. So there are other considerations that go into timing when to pick that don't have anything to do with bricks or acidity. And I remind winemakers all the time, as a winemaker, it's not just tasting, it's not just smelling, it's looking, it's touching, it's feeling, it's listening. Use all your senses. That's the glory of winemaking, is that you do get to use all your senses. So with all that being said, let's get down to brass tacks. What I call the Midwest winemaking high acidity blues, or Houston, we have an acidity problem. Excuse me. What I want to make you all aware is that you have many different techniques and methods to deal with high acidities and low pH. It's best, I always advise people to use a combination or a variety of methods. Use all the tools in your toolkit, not just one big hammer. We're going to talk about methods of acidity manipulation that work quantitatively, that literally reduce the total acidity. And then we're going to talk about a few techniques that can help reduce the perception. They may not bring the TA reading down, but they make the wine more approachable. Easiest one, the one that everybody, home winemakers start with, and you first learn about in deacidification, is to use what I call, under the broad term, carbonates, whether it's calcium or potassium carbonate or potassium bicarbonate, etc. These are magic powders that will absolutely lower your acidity. But having worked with them both in must and in finished wine, I don't like these darn carbonates, or I don't like them to use overused or used very much. Overly used or applied too late, they can certainly mute aromas and flavors. They can, out west in California, the old winemakers call it plastering. Well, that wine's got too much acidity, we'll just plaster it with a carbonate. And it can have some deleterious effects on the wine. It kind of flatten the palate, sometimes even give it a chalky feel on your tongue if it's overused. However, it is a tool, and I know some winemakers very successfully use a small amount of these compounds. I encourage people to try, if you're going to consider using these carbonates, use them early. Adjust your must early, get them in there before fermentation. That seems to integrate the effect better. Overall, I'm a big believer in trying to adjust your must before adding yeast, whether it's adding carbonates or adding sugar or adding acid or whatever things you can add to adjust your must. Try to get that adjusted before you initiate fermentation. In general, 1 gram, they all slightly different, but in general, 1 gram of a carbonate will lower your total titratable acidity by about 1.5 grams per liter. That's handy. I don't advise using more than about a gram per liter. People use as much as 4 or 5 grams per liter. And again, I thought the effect on the wine was negative. And always, no matter what you're doing in winemaking, when you're adding something for the first time or something you haven't used before, bench trials, small scale bench trial manipulations help. I get a lot of hard looks sometimes on this technique, what we call amelioration or adding water. I want to assure you that adding water is not cheating, and it's not illegal, and it's not rare, and it's hardly ever done, I can assure you. As a former West Coast winemaker, and I worked for Gallo for 3 years in a Gallo winery, adding water is a normal course of events. And I hate to overplay this point, but of course, every gallon of water you add is a gallon more of wine you've made. So without question, adding water will lower your total acidity and raise your pH at different amounts. There's different buffering levels in wines, but no matter what, adding some water will help lower acidity. If you don't know this, of course, you never want to use any chlorinated water, so the quality of the water is important. I say small additions work without dilution of the flavors or aromas. Though I have heard of people adding up to 15% by volume of water to a very, very early picked Frontenac Blanc must. And they ended up making a balanced wine out of it, and apparently it was not thin or diluted. It still sold very well and was a balanced wine they were able to work with. In general, amounts 2 gallons per 100 gallons will reduce your TTA by about 0.1 gram per liter, which is neat. If you've got real high acid and low Brix, add water and sugar. Again, it's not illegal out here in the Midwest. For anybody who doubts that it's legality, I have the Code of Federal Regulations citation there on the bottom, which you can read for yourself. And I don't know many grapes here in the Midwest that are below 5 grams per liter. All righty, moving right along. Probably the most important way for you to lower acidity is in your choice of yeast. There are some really neat yeast strains that metabolize malic acid and convert it to ethanol, which is a really neat trick. As if the yeasts weren't cool enough converting sugar to alcohol, they can convert malic acid to a little bit of ethanol, which is, like I say, I think it's a neat trick. Y'all got to stop and say a prayer for yeast someday. Where would we be without yeast? I think I just read somewhere that every drop of fermenting wine has something like 2 million yeast cells in it. My mind. All right. The yeast known as C by Lalvin can lower your total acidity by about 40%, has a pretty decent alcohol tolerance, and a lot of people use it for sparkling wine production. Here's a workhorse Midwest yeast for fruit and red wines. I use this all the time. I encourage people to use it all the time for grapes like Frontenac. I say it's good for reds and fruit wines. It's nothing wrong with using 71B in white wines either. Do keep in mind that it has a slightly lower alcohol tolerance, only up to about 14%. So keep that in mind if you're concerned about your fermentations stopping before they're finished. Yeast, not a lot of people know about SVG from Lallemand, also lowers TA considerably. Great for aromatic whites. I know people use it very with good effect for Le Crescents. Has a relatively good alcohol tolerance. A variety of other strains as well besides those 3. I haven't used this Morven B, which is an Australian yeast, but they claim that it converts up to 56% of malic, which is a lot. It has a high alcohol tolerance. Like I say, I haven't used it yet. I need to get a sample of it and use it. So far, I've only been able to find it through GW Kent, and they seem to always be sold out of it. And Vintner Vault in Paso Robles, California has it on their website. And then the other three yeasts, exotics, opal, and GRE. GRE, by the way, is a wonderful yeast for Grenache. I think that's even where it gets those initials, but it also can lower acidity. Of course, malolactic fermentation will lower acidity. Maybe you don't want to put your crisp aromatic white wines through malolactic fermentation. A quick note about that. 2016, I made some Lacrescent research wines for the University of Minnesota, and I did some yeast trials, and then I split those up and did some malolactic trials on Lacrescent. And I always thought, you know, one of the great things about Lacrescent is all of its high, beautiful floral aromatics. And I thought, oh no, ML is going to really mute all those wonderful aromatics. And it did, but not as much as I'd feared. And another thing, one of those lots I made would not finish malolactic fermentation. It only partially did it. And, you know, there's nothing wrong with partial malolactic fermentation in whites. They do it a lot in France with Chardonnay. They can get it, go through, they can split the volumes out, put one half through, or a portion through ML and a portion not through ML, and then blend the two portions back. You sort of get the best of both worlds there. You get some of the creamy, richer texture, but still retain your aromatics, your vibrant flavors. So you can actually use MLF for some of these aromatic whites, but in general, it's used, of course, for reds. And I just remind people, if you're not comfortable with malolactic fermentation, I always think green apple to milk. It's a bacterial fermentation that converts the harsh malic acid, symbolized by the green apple, into a softer lactic acid, sort of the lactic acid you find in milk or cream or butter. My first harvest here in Minnesota, I had some Marquette, a couple of batches of Marquette. And despite all my efforts, I thought the acidity was still too high. And I gave a call to my predecessor at the university, Katie Cook. Katie now works for Scott Labs. And she said to me, oh, well, have you put your Marquettes through cold stabilization? And I said, oh, Katie, you don't put red wines through cold stabilization. And she said, Drew, you're not in California anymore. And there's absolutely every reason in the world that you can put red wines through cold stabilization. And just like with white wines, excess tartaric acid can and often will precipitate out as crystals, as wine diamonds, as we call them. So you can cold stabilize your high acid red wines, and that will lower the total acidity. I remind you all, if you're cold stabilizing white wines, you want to, before you stop stabilizing them, you should get them tested, get them conductivity tested to ensure that stabilization is complete. The staff at the Midwest Grape and Wine Industry Institute at Iowa State University has a lab. You can send them samples and they can check that for you. Do keep in mind that cold stabilization, traditional cold stabilization does take a lot of power. You got to have a cooling system and jacketed tanks. So it can be an expensive proposition, but once again, it is possible. It's not a silly idea. You can cold stabilize your wines simply by putting them outside. You got to be careful not to freeze your wines. You may not want to put them out when it's 10 or 20 below zero or something, but certainly you can put them out when the temperatures are between freezing and, say, 10 degrees or so above zero. And that, in general, can cold stabilize your wines. I'm sure there's some folks out there who do this all the time. And yeah, I'm looking at these smaller tanks. It would be hard to use the weather to cold stabilize a 3,000 gallon tank. I agree. So there you go. As I mentioned earlier, one of my favorite ways to lower acidity is simply by blending. And it's Aude, how am I doing on time? How much longer do I have left?
Yeah, I was just saying 5 minutes remaining.
Excellent. Perfect. Thank you, everybody. So it's not rocket surgery. It's a simple fact that blending low acid wines with high acid wines will lower your acidity. I'm a big believer in blending. I know early or young or newer winemakers tend to be purists and want to make just single varietals. and that's a fine thing to want to do. But sometimes the business and the market calls for blends. There's a reason you have a 75% varietal label. You can put 25% Cabernet in your Marquette, still call it Marquette. Blends do sell. Best-selling red wine right now is Gallo's Apothic Red, and that's a blend. And sometimes by acknowledging wine blending, it's a great way to help marketing your wine. People are excited about that. They like to imagine the winemaker as sort of a scientist, mad scientist, coming up with these. Those are the ways that you can lower acidity. I'm going to talk about a couple of ways here to lower the perception of acidity, just in case you weren't aware. Barrel maturation and extended sur-lie aging can work to reduce the perception of high acidity. This autolysis reaction that happens with the fine yeast lees in barrel and in smaller tanks can not only build complexity in your wine, but can help soften the texture. And aging wine in barrels encourages malolactic fermentation. Again, these are textural reductions, not quantitative reductions, but barrel aging, neutral barrel aging is a great way to do this. And here's a gentleman with glass heads on one of his barrels to show you what it looks like to stir those lees up. Sweetening is probably the most often used technique for counteracting high acidity. And I know you're probably all aware of that, but while we're on the subject of sweetening, I want to remind you that there are 4 ways to sweeten. Of course, you can add sugar, but there's 3 other ways besides that. Stopping fermentation early, either with your yeast choice, a low alcohol tolerant yeast, or with sulfur, addition of sulfur dioxide, cold temperature crashing, sterile filtration. These are ways to stop fermentation early. In Germany, they use a Süß reserve, a sweet reserve that'll save out some of the original unfermented juice and add it back prior to bottling to give it that natural balance of glucose and fructose that was in the original must. And grape concentrate is a wonderful way to back sweeten wines. You got to check your source. You want fresh, clean concentrate produced by somebody who knows what they're doing, and it wants to be kept chilled or even frozen. Occasionally, there are stories of a very harmful yeast called Zygosaccharomyces that can infect concentrates. So, yes, you can use concentrate, but make sure it's in good condition. So, I encourage you, again, to use bench trials. Always try to do your manipulations, your additions, your blends on a small scale before applying them to a large scale. I can assure you that very tiny percentages of changes in sweetening or acidity balance or all these sorts of things small changes can make huge effects in the finished wine. I always encourage people to do scaled additions and test bench trials. And always keep the control in that group. Helps to maintain your sensory objectivity. Choices, use them all. Don't just use 5 grams per liter of carbonate when you can make some good choices about yeast and cold stabilization and blending and even watering wine. All right, I know I'm getting close, so I will just acknowledge quickly University of Minnesota, where we're driven to discover. Supervisor Matt Clark has a great breeding program, John and Jenny Toole out in the vineyards, and all the other grad students and extension folks as well. And there I am in Santa Barbara 20 years ago, not even thinking about Minnesota. Thanks so much.
Thank you very much, Drew. That was a great presentation. That was great to have an overview of all the different techniques you can use in your winery. So even if you're not planning at using those tools this year, maybe in the future you will need those information. So now that's my pleasure to introduce Dr. Misha Kwasniewski. Hopefully I'm pronouncing correctly your last name. So I'm gonna introduce you. So Dr. Misha Kwasniewski is the Assistant Research Professor of Fermented Beverages at Penn State. Before taking his position at Penn State earlier this year, he worked at University of Missouri, where low tannin and high pH, high TA wines were among the chronic problems faced by producers. His research focuses on understanding and remedying problems faced by wineries, distilleries, and breweries through the lens of analytical chemistry. He has also worked extensively to elucidate the relationship between growing conditions and flavors in fermented beverages. So today, Dr. Kwasniewski is going to present about or talk about the different pH, total acidity trouble that we have in the Midwest mostly, and about a new tool, or kind of a new tool, that is the ion exchange method used to deacidify wines. So Misha, I'm gonna give you the presenter role.
Excellent, thank you.
Yeah.
And we'll see how quickly I can do this, and if I actually...
That's fine, you have 30 minutes, and just a reminder to all the attendees, feel free to ask or type all of your questions either in the chat or the Q&A box. Thank you.
All right, am I up successfully?
Yep, all good.
All right.
Thanks.
Wonderful, thank you. So I'm going to talk about a process that really before that, moving to the Midwest and being part of Midwest winemaking and winemaking problems and research, I had no idea was even a reality. And that is sort of the worst situation where you don't just have a high pH or a high TA, you have both. And so lots of those great solutions that Drew outlined sort of go out the window. I'm going to very quickly go over a little bit of what Drew already introduced so I can sort of speed through this, but I want a little bit of the chemistry to be on your mind, because unfortunately, for those of you who are maybe not interested in the chemistry, to utilize and really understand what's going on with cation exchange, it really becomes a matter of understanding the pH-related chemistry. And so one of the critical points that was brought up earlier is this idea that pH is a log scale. And so that means to change from pH 7 to pH 6 or pH 4 to pH 3, we aren't trying to change we're not trying to change from 1 gram per liter to 2 grams per liter or something like that. We're trying to cause a change of 10 times of the hydronium ions. So anything that we need to do is going to be sort of by its nature, when you're trying to change pH, very extreme and take a lot of change to that wine in a way that things like, for instance, carbonate, you would have to use a lot to change things around, although that would be more going the other direction. But same thing with trying to add acid. You would need to add a lot of acid to bring a wine from pH 4 down to pH 3. So, as was mentioned earlier, pH is critical from a standpoint of biological stability, color reactions, color stability, the actual nature of color. As you get higher and higher to the extremes of above pH 4, some of those anthocyanins can take on a less desirable color. So there's really some benefits to adjusting it just from that. Also, for a whole slew of reasons, the oxidation rate is higher at a higher pH, and that we also care from a standpoint of protein stability. So generally, as we get the pH at extremes of extremely low, then it's actually easier to precipitate proteins, which is sort of counterintuitive, but that extreme nature makes it more readily that the unstable proteins fall out versus if you're closer to neutral, where they're likely to sort of linger around and only pop out when your consumer decides to open a bottle of your wine. Titratable acidity, or frequently referred to it as shorthand as TA, is important for taste, and I think also has a lot of outsized importance in the winery because it's something that's easy to measure. It encompasses a lot of things, but it also isn't really demonstrating the underlying organic acids or cations or everything else that's going in to make this pH acid relationship happen. It tends to correlate quite highly with wine with total acidity, but they aren't one and the same. Really sort of a seminal work of wine research. Roger Bolton has a beautiful paper where he demonstrates the relationship between total acidity, which total acidity is the sum of tartaric acid and malic acid and acetic acid, and to a lesser extent, succinic and a few others. Whereas titratable acidity is either the amount that it takes to titrate your wine with NaOH up to pH 8.1, or you can calculate it if you know the amount of, the titratable plus potassium plus sodium. So total encompasses both the titratable and the cation extent, whereas titratable is just a matter of the actual amount it takes to titrate your wine. So total, in a way, is very useful, but it's also incredibly hard to measure because you need to actually have expensive specialized equipment like a HPLC to measure it. Just to reiterate this point, that if we looked at sort of a normal wine that could have a titratable acidity of about 6 grams per liter, accounting for what that 6 grams per liter is also being effective and buffered by the potassium content in that wine, it could actually have a total acidity of close to 8 grams per liter. And so just from that standpoint, as you're trying to think through, additions and manipulations, that even if this was 50-50 tartrate and malate for total acidity, that's a much different number than if you're assuming your 6 grams per liter is 50-50 and you have 3 grams of each. So this problem of high pH, high TA wines, I'd sort of heard of and wasn't, I thought it was a boogeyman that happened on occasion. And then my first year doing research wine at University of Missouri, our Norton came in at a TA of 13 grams per liter and pH of 4.1, which is terrifying, especially when you're doing small lot experimental vinifications. You have something over pH 4 and you pretty much might as well throw it out into the drain right as you're beginning the process. So right there and then I knew that we needed to start understanding and addressing this, and then those, especially in Missouri and Arkansas and Iowa before me, that had run into this problem and had characterized a lot of what mattered. And cultivar certainly is one of the critical things. If you have a variety that wants to have a high TA and high pH, then it's going to, most years, no matter what you do about it. Also incredibly important is the amount of water uptake. So as you irrigate more, or even if there's water and the vines transpire a lot, you're likely to have more potassium uptake. And as you have more potassium uptake, you have higher and higher pHs. And if your fruit also has high organic acid content, which many hybrids have, especially starting out, very, very high malate content, you're going to have a problem. There's other factors such as fruit maturity and soil and vine shading, and a lot of work has been done trying to control this in the vineyard through either changing rootstocks or adjustments in canopy manipulation. And I would say probably if you sum all of the practices, keeping the TA the same, you could probably pull a wine whose pH was 4 down to 3.8 or 3.9. So there's some work that shows that there's significant differences, but not really meaningful from the standpoint of wine stability and safety. So the reason for this high pH is that, predominantly calcium, sodium, and potassium compete with the hydronium or H+ ions, which that is what causes pH. And so those of you, especially I think, that are growers deal with this a lot with the idea of soil ion exchange. And so if you've got more of one, it just tends to be more present in the solution. And so if you've got lots of potassium, then it starts to outcompete the amount of hydronium ions that are out there, and then that drives the pH up. The problem is that the solutions for either dealing with the high pH or high potassium are sort of in opposition to each other. So if you added tartaric acid, that's a great way to lower the pH. However, it's also a great way to very directly add to your TA problem. There's been some work that's demonstrated that you can also use electrodialysis. And if you find that cation exchange is complicated or cumbersome, this is definitely far beyond that. But who knows? I think especially as the industry develops, there's probably room for somebody operating a custom mobile electrodialysis machine to directly strip out cations. Cold stability or cold precipitation can help. The more potassium you pull out of that wine, the less it is going to be buffered. And as you pull potassium, you're also pulling tartaric acid out. So that's great. But again, if you're dealing with extremely high TAs and high pHs, it's probably not going to do what you want it to. Another solution that's been used is double salt deacidification, which is complex, and I think in the real world has varied results and is really quite ineffective when you're trying to deal with really high malate varieties. So I ended up looking at ion exchange resin as the solution, and it has actually been approved for wine going back to the 50s. Again, TTB explicitly approves it, essentially, as long as you don't drop your pH below 2 or something absurd. And it has the benefit of not only swapping out hydronium ions for potassium and changing that pH, but at the same time, it's an effective way of cold stabilizing your wine. It's also essentially instantaneous. In a matter of seconds, you know what the results are going to be. And the resin itself is very low cost. However, as I got into this, there really wasn't any easy, small to medium-sized winery way of doing this. There's giant, expensive units that can be used and adopted from like water treatment facilities. But when it comes down to it, as you'll see, it really isn't that complicated a process in comparison to some of the things that you're already doing in the winery. What does the work is cation exchange resin beads. And so these are little round resins that are inert or theoretically inert, and I'll get into that a little bit, that have active sites all around the outside. And these active sites have, in the case of cation exchange resin, an affinity for cations, so potassium, calcium, or H plus ions. And if you want to directly change what is in your water, or water or wine, but these are frequently used in for water treatment, you load the resin with something that is preferable. They're even used in home water treatment, frequently loaded with sodium ions. In larger facilities, they use the hydrogen-loaded ions. So on each one of these little active sites, you've got an H plus that will go into solution. And then for each H plus that is lost by the bead, you're going to have an affinity for whatever cation is heavily present. In the case of wine, we're going to predominantly be pulling out potassium. And so it's really a twofer that we're removing buffering capacity, but we're also directly influencing that pH by driving it down. Just to look at this a different way and how it actually works flowing through this resin bead, this resin bed. If you've got high potassium-rich wine flowing into resin that initially is covered just with H plus ions, some of the H plus are displaced and go out with the treated wine, and some of the potassium ions are being sequestered. How efficient and complete this reaction sort of depends on how fast you're flowing the wine through. And on the extreme end, I've known some wineries that in fact just put a bunch of resin in a sack and stick it in the top of a wine and then pull it out after like an hour, knowing that it's done its work. The setup that we work towards designing was really trying to make it so winemakers had a lot of control, so that it wasn't just a matter of, well, let's dump this in and see what happens, but that if you've passed the wine through a exchange bed, and it can be anything from custom built units that can be used for water to things that I'm going to show you how you can build your own that can do up to really easily 1,000 gallons or more per pass, is that you're mainly using equipment you already have in the winery. And so you pump from one tank using a standard, wine pump through a resin bed, and then pump into a holding tank. You don't necessarily have to pump or treat all of your wine to get the effect that you need. If you are able to drop a portion of your wine to a good blending element, you then can, be able to use that, to be used that for blending. So some real world numbers of what we ended up finding as we were doing trials on sort of lab and small winery scale, that a wine, in this case, it was Chambourcin, that went in with a pH 4.1, relatively low TA in this case, but wine research doesn't always hold for exactly what you need, but it went through the resin bed. The potassium ions dropped by nearly half, from 2,200 milligrams per liter down to about 1,300 milligrams per liter. And we were able to drop that pH all the way from 4.1 to 3.5. To look at this a different way, not all of, well, so as you're pumping, not all of the reaction happens instantly. And also there's some different preference as far as different resins between potassium, calcium, and magnesium, as well as you're shifting a bunch of the equilibrium. And so immediately with this wine, after introducing it, you have a shift from, in this case, a wine that was 3.9 down to an incredibly high or incredibly low pH of 2.5. And then that starts to creep up as all of the resin active sites are utilized until we get this final value where basically the pH of the wine is the same as what we started. However, we've shifted the cation content quite a bit. What we put together is basically we use a spool of sanitary tubing that you can buy in any number of diameters. Now, especially with the small distilling being quite in vogue, there's just lots and lots of options as far as diameter. But this setup, we were using 3-inch spool, and the sight glass was really just there to show what the resin ends up looking like in that tube, with a gasket with a screen. Now, unlike trying to push through a filter bed or something, we're not trying to pack this spool so that there's a lot of back pressure. We're actually trying to design the system in a way that the wine flows really, really easily in. So we generally only fill the spool up about 2/3, and then slowly pump the wine through with all of the resin being trapped. Now, again, there's as many different ways of attacking this as there is really winemakers and ways that they can think through utilizing their equipment. I've known some wineries that have done this effectively by dumping the resin directly into a tank and then pumping out through a mesh gasket or some sort of filter. The one warning I would have of doing something like that is the little resin beads are really, really annoying and get everywhere. So you, as best you can, want to control how you make it that they don't end up everywhere and in your trench drains, and that you're seeing these little shiny flecks for the rest of your winery's life. Back when I was in Missouri, Les Bourgeois was nice enough to host a demonstration, and we did a couple of 200-gallon per run batches. Now, that 200 gallons, again, you could scale it up to do more per run, but was actually enough that they could then blend in another 300 gallons to have a full 500 gallons of wine that was going into barrel at the pH that they wanted. And so thinking of this as another tool in your toolkit to create wine for blending to get it where you want is an option, not necessarily that you're going to all of a sudden have to treat every wine fully to drop that pH. Let's see if we can get this to work. So just to give you an idea what this sort of looks like, you can see that this really is not a fast pumping system. We're just slowly and easily trying to pump from the bottom up through the resin bed, allowing the wine to fully interact. And then as it comes out, you've changed the pH and changed the cation content. The main steps to effectively doing this is, first, you need to rehydrate the resin. So it comes somewhat dry and probably only about 50% to 75% of the final volume. And so if the first thing you do is pack dry resin into something, it'll keep expanding on you to the point that it will really close down the system. So first, rehydrate it for a couple of hours. I recommend using like 100 ppm SO2 solution, just to make sure that you're keeping everything nice and sanitary throughout the steps. Then rinse the beads, rinse the entire system. So run the system through with water just to remove any particulate or any extra sanitizer might be there. Slowly pump the wine through the resin bed. You shouldn't have really much back pressure at all. A variable pump is ideal, but you can also construct a bypass so you can keep that pumping slow and steady. And then just monitor the pH as it comes out. And once you reach the starting pH, you're done. You've reacted all of that resin. You can rinse the resin and actually reuse that resin over and over and over again, really as much as you can keep it nice and clean and sanitized. So if you think of it as like a really, your nice final absolute filter that you can probably get several runs, or if you're a small winery, you may be even getting several seasons through. The same thing with the resin, that initial investment, you can recharge with a strong acid, which I've outlined the calculations on a sheet that I'll make available to anyone who's interested, and keep using that resin over and over again. And then store in an SO2 citric solution for the next time you want to use it. For about 500 gallons to treat, it's going to be about $150 worth of resin. But as I said, you can do that over and over again. So if you get 2 batches out of it, you're now down to $75. If you get 3 batches, $50, and so on and so forth. So it really can be a very cost-effective way to adjust both your TA and pH. And you can get about 50 bed volumes, or for a given cubic foot or liter, so for every liter of resin, you would get about 50 liters that you would treat completely from that wine. In practice, I think the most important thing that was said earlier is start with benchtop trials. This is immediate. So you put in different amounts, you see what the effect is, not only on the pH and TA, but also any other effect that may be happening with your wine. And if you don't like what it's doing to your wine, don't use it. It's just not the option that's going to work for you. So as you're changing the pH, also remember that you're changing the amount of SO2 that you're going to need. So dosages should be adjusted. And there's some questions that I'm very briefly going to go over related to what it may do to wine flavor and other characteristics. One of which was sort of the last steps of one of my graduate students that was quite surprising. We found with very high additions, we actually got a reduction of alcohol, which is sort of a nice side use for this. Under normal conditions that I would say that you would be using for adjustment of pH, you're always going to be under this about 5 grams per liter, 5 grams of resin added, which is sort of a meaningless unit right now, But so you might see 0.1, 0.2 reduction of ethanol. However, it is potentially an option to, with no energy, actually sequester ethanol and reduce that after the fact. Likewise, there's some shifts that happen with some of the organic acids and some flavor compounds, all of which we're now identifying is due to equilibrium, not with the actual active sites, but the resin itself, that sort of acts as a semi-permeable membrane. And so if it was hydrated with water, or you ran through a water or a wine that was very low alcohol before it, and then did a high alcohol wine, more of that alcohol or organic acid or flavor compound is going to be sequestered by the resin. In some cases, this can be a good thing. It can pull out a little bit of things like volatile acidity. But by and large, I would say these are sort of secondary impacts that if you like the impact that it had on your bench trial, then you're going to be happy with it as an option for your winemaking. And then just lastly, we did some work with sensory, and there was at no point in the descriptive analysis that the cation exchange ever enhanced any negative attribute. In fact, it tended to reduce some sulfur aromas on some of the wines. And in some cases, there was instances where it actually increased things like fig and cherry and Norton. But for the most part, it really didn't have a lot of impact that could be picked up from a sensory standpoint. So with that, thank you for listening. Also, I have to thank the Missouri Wine and Grape Board, as well as the Missouri Marketing Wine Research and Marketing Council that helped fund this while I was there. Brian Wayne, Megan Wesolensky, and Joe Beretta are all grad students that have worked on this project over the years, as it went from something that was sort of a quick and easy idea and got more and more complicated. And sort of the take-homes are it's effective. It can have some results on quality, some of which is actually positive, and there's a pretty modest cost associated with it. So with that, I will stop so we still have some time for questions.
Thank you very much, Misha. Right on time.
Excellent.
Okay, so thank you for your great presentation. That was maybe a little bit too much science for some of the attendees, but that was very interesting. That's always good to see the different options people can have. So on that, we will start the question and answers. So we will start probably with question for Drew. So the first, but Misha, if you want to help answering, or if you have opinion, feel free to answer as well.
Okay.
So you mentioned, so Drew, you mentioned that lower pH wines age longer and higher pH wines are more susceptible to spoiling. Is there an ideal pH?
Oh boy. No, winemaking is not recipe driven. A brewer has a recipe, and a brewer can create the exact same beer with the exact same chemistry and flavor profile every day of the year. In winemaking, we get a different crop every year, and every year is different. And the same grape grown in the same vineyard is going to have different Brix, pH, and TA every year, depending on. So the short answer is no, I don't really have an ideal pH in general. White wines tend to be on the lower part of the scale, let's say 3.1 to 3.5, and reds tend to be on the higher part of the scale, let's say 3.35 to 3.7 or something like that. These are generalities, but every wine is different, just like every year is different.
Especially as everyone mentioned, like the pH would be very different if you're talking about a vinifer wine or if you're talking about a hybrid wines. So I think there is no good real answer.
I'll go a little bit out on a limb and say that while there may not be any good answer, there's bad pHs from a winemaker's perspective. And if you get above 3.8, 3.9, you're going to be scared. So but you want to keep it below that. And you get below pH 3, and I've had some beautiful, dry Rieslings that can handle that, but you're sort of in a danger zone. And so you want to make sure that you're on point with all your winemaking.
Mm-hmm. Yep.
I would certainly agree with Misha. Absolutely. And with point as well is that general vinifera tend to be higher in pH and lower in acidity, and these bitter-specific hybrids tend to be higher in acid and lower in pH. Thank God I've never had to deal with a Norton that came in at 4.0 with, what was that, 12 or 13 grams of acid? I think I would kill myself, but not anymore. Now I have an answer. I would call you up immediately, Misha, and say, where the heck do I buy some resin beads?
Well, as a researcher, it was actually, oh, wonderful. I've got something to do here.
Exactly, for sure. Yeah. Was there another? I had a question on the chat. Oh, did you see that?
Yeah.
So
regarding blending?
Yes.
Yeah.
Okay. I'm a little confused by the question. Let me just say, when I was talking about blending a low acid wine with a high acid wine, and I used the example of Cabernet and Marquette, and again, these are generalities. In general, when you order, and I'm talking about ordering grapes from the West Coast and making the wine here in Minnesota and making the Marquette here in Minnesota or wherever you are, I'm talking about blending finished wines. And I think it's inescapable that almost any Cabernet is going to come in with a relatively lower acidity, let's say, maybe only 4.5 or 5 or 6 grams per liter at the very top. And it's going to have a relatively high pH, let's say above, 3.5. And in general, Marquettes won't have that high pH and, that high of a pH or that low of an acidity. And so adding one to the other will lower the overall acidity. Yes, there are exceptions. And I've also just this year or the last 2 years run into a couple of events with Marquette where the Marquette was picked and crushed and the initial pH came in at say 3.3, which is rather normal. And then at the end of fermentation, that pH had jumped up to 3.9 or 4.0, which was surprising. And again, this is something that I think Misha could probably speak to in terms of some viticultural practice, excess calcium or potassium or something. But anyway, to answer this question and stop beating around the bush, in general, I'm talking about you can take almost any red vinifera finished wine and blend it with almost any red hybrid, and you will make a more balanced wine. The Cabernet and Marquette issue, of course, one of the other things is that Marquette tends to be lower in tannin, and Cabernet tends to be higher in tannin. And when I've done blending of wine after fermentation, right before I was going to do cold stabilization, I enjoyed the effect. It made a more balanced wine. Those tannins were retained better. And those wines, go ahead.
Can I ask you the same question? But if you don't have any vitis vinifera to do the blending, what would you suggest? Or is there a specific wine that you can blend, both coming from hybrids? Like if you want to lower the acidity of a Frontenac, what would you recommend? Do you have a hybrid variety that you will prefer to use to blend and reduce the acidity? Or
I used the example earlier where I made a La Crescent based blend, and the La Crescent was low in pH and high in acidity. I blended it with some of the Swenson varietals, with St. Pepin, with a little Brianna, with some Prairie Star, and all of those three had more moderate pH, more moderate acidity. And I blended it, that wine ended up being about 55 or 60% La Crescent and 40-45% a blend of these other Swenson varietals, and it took the edge off. It balanced it out, and it really added to the complexity. That's the thing about blending is in blending, 1 and 1 doesn't always equal 2. 1 and 1 can equal infinity sometime in blending. You can't put your finger on why you like the blend parts. It doesn't always happen.
A plug for the program that you're involved in. I think that exactly demonstrates why breeding is necessary. That a lot of the characteristics that go hand in hand with what we want for things that can handle the cold and have disease tolerance also are coming from varieties that are incredibly high acid. And so to decouple those is really a Herculean feat of great breeders that's going to take a bit. But I think the indications are that it's there, that what there might be in 5 or 10 or 20 years, it's going to be a whole different game for Midwest winemaking.
I agree.
Yeah.
And I must say, in all humility, we're not the only institution working on this problem. There are a variety of private breeders out there who are tackling this. Of course, Cornell University has done a lot of research. It doesn't get quite as cold there as it does here in Minnesota, but they are also working on this problem. And Washington State University as well. So there are folks working on it, and as you say, we're learning more and more. Genetics, the field of genetics and what's called marker-assisted breeding is expanding all the time. For example, we've some work a graduate student did at the University of Minnesota. His name was Soon Lee Tae. He's now at Washington State University. He was able to locate 4 spots or loci on the grape genotype that signified resistance to powdery mildew. And we have already been able to adapt that technique and look at our thousands and thousands of new grape seedlings every year by taking a tissue sample of the leaf and looking at its genome and saying, these 9,900 do not have that mildew resistant gene, but these 100 do. And so we'll throw those others on the compost pile and just work with these 100. So it may not speed up the process from start to finish to get a new grape, but certainly it creates an earlier bottleneck that allows us to make better decisions earlier on what new seedlings we should keep working with.
Yep. Thank you. So I have a question regarding the cation exchange. So your presentation, Misha, can you talk about the cation exchange and its stabilization effects?
In relationship to like potassium or just in general? So
maybe just in general.
Okay, yeah. So I mean, one of the things that's a very nice side effect is you are actively removing cations. And so from a tartrate stabilization standpoint, it's effective, and it actually is about the only solution for calcium tartrate instability, which rears its head every so often. I 3 years ago had a winery in Missouri that came to me with this weird problem, and as we got further and further into it, it was like, oh, I've read about this as a thing but never actually seen it. It also, through lowering the pH, you're going to have a much more microbially stable wine. And so, you know, even and this is true whether you're making the wine that you want or not, but a wine that's at pH 2.9 is always going to be more stable than one that's at 3.5. That it's just, it essentially is in stasis, and you as a winemaker can sleep better. People may not want to drink the wine, but you know that wine's going to stay with what it was. So it's a really, I think, important tool from that standpoint, that if you know that, you know, you've had problems with bread in the past, or SO2 management is something that isn't your friend, bringing that pH down puts you in a space that you're a little bit safer throughout. I've actually sort of wondered, you know, the effectiveness for people that are looking into distilling brandies where you can't really use SO2, that if you can drop that pH way, way down, you've got something that's very stable until you finally get to the point where you can distill your product.
Yeah, agree.
So just a quick follow-up. When is the proper time to do ion exchange? I assume you don't want to do an ion exchange on wine A and then on wine B and then blend them. Is this something that happens before blending and before cold stabilization, or is this something you could do the day before bottling?
Yeah, I had the same question.
So definitely you want to do it before final cold stabilization, because it's going to throw everything out the window, probably make it more stable, and it's not worth going through all that cost and effort to find out that you in fact didn't need to even cold stabilize anymore. I would say it can definitely be done before blending. What we were not effective to do, and to a point in Drew, your presentation, I completely agree that any intervention you can do before fermentation is ideal. And we tried that and made a horrible mess. We're not able to figure out any way, including pressing off a red to have its juice by the side and try and treat it and reintegrate. It just, it may be possible with, you know, a team of engineers helping you out, but as far as coming up with a cheap, easy way to do it with existing equipment, that wasn't happening. So basically, once it becomes wine, and any time before you're about to stabilize is an all right time to do that. And mostly what I have seen wineries using it for is before it goes into the barrel. So then you've got something much safer that's sitting in that barrel aging.
Yeah. And so we have a question related to where would we find the resin beads?
Your local Culligan man, online. They're actually very readily available. What you just need to check as you're talking, especially if you talk to a salesperson, they are first going to want to sell you the sodium loaded beads, because that's what most homeowners use. But they all have the hydrogen loaded beads available. It's actually quite widely available.
Okay, good. Thank you. I don't think we have more questions, but if we didn't answer any of the question, or just if you have a question, you still have like 6 minutes to type it. That's more a comment that we have in the chat box. Or we have 2 comments. So the Swenson cultivars have common pedigree in different combination, and when you blend, as Drew suggested, it's like a family that sings together, compatible tones, but slight difference bring a fuller sound. And then quite often, Midwestern wineries are shooting for a particular pH related to ripeness. Does this allow a grower to harvest earlier and avoid some of the bad characteristics when fruit ripens further? That's a question. Misha's technique.
So the problem, I suppose, is that the cation exchange wouldn't help you with removing malate. And so it could be a tool. So if you thought in a multi-step process that you neutralize some of the malate, or you were going to go through ML on an early grape variety so that you hadn't really gotten that full descending on the curve, that then you could still adjust, have a unacceptable pH, run it through cation exchange, and then bring everything back into focus. I have not played around with that, but I think theoretically, as part of a multi-step, winemaking sort of package, it probably could work.
Okay. So we just have another question in the Q&A, but I think that's similar to where we can find the resin beads. Where can we get the paper on recharging, caring for the beads? So that's related.
I can share it. Probably easiest. This is sort of still in the beta phase, so I definitely am looking for any feedback for anyone who starts working on it, and have my email front and center on this sheet. But it takes you through all the steps, including recharging. And I call it a brief, but it's probably, I think, about 4 pages of fairly dense instructions. But I could make it available to either individuals, or is it easier that I could have it, that you could post it somewhere? What seems easiest?
You can, so if you are looking, so the attendees who are looking for information, they can reach out to us, and then we will spread the information to you, Misha or Drew. Yeah. So that's it for now. I think we don't have more questions. Just to finish on, so first I would like to thank you both for your great presentation. Then I want to explain you that we will have another research and winemaking webinar next week at the same time, so 3 p.m. Central Time. That would be on malolactic fermentation. So stay tuned to have a follow-up on the acidity using malolactic fermentation. Then we will have another webinar on November 10, same time, on filtration, followed by in November 24 on barrel maturation, and then December 8 on bottling and closures. So stay tuned for those next webinars. If you have any question, feel free to contact us for any question. So at watrelot@iostate.edu or to dhorton@umn.edu. And remember to take like few minutes to complete the survey you're supposed to receive by email regarding how we did today and what we can improve. I think I saw a comment or a question chat requesting the beads presentation. Okay. And will the webinar be available to view again later? If you need to have access to the recording, feel free to send me an email, and I will create a file, like a link, to have access to the presentation. On that, I think we're good. What else to say? Thank you, everyone.
Yeah, thank you
everyone
for having us.
Yeah, thank you.
All right. Bye all.
Bye.
Everyone.
Bye.